So in this segment I'm going to talk about Krebs cycle, which comes right after glycolysis and deals with acetyl coenzyme A. Unlike glycolysis, Krebs cycle doesn't happen in the cytoplasm of the cell, but happens in the matrix of mitochondria. First of all, we have acetyl coenzyme A right here, which gets attached to the molecule of oxaloacetate that we have right here. So what happens that from oxaloacetate that has 4 carbons, we get citrate that has 6 carbons. But it's a cycle, so we will have to get back to a 4-carbon structure in this process, and we will see how. So when the acetyl coenzyme A comes, it's with the help of water, which we have right here, and it gets attached to this carbon right here, and it forms citrate, which is salt of citric acid. Very important thing about citrate is this hydroxy group, which makes it tertiary alcohol. So to the carbon here, we have attached three carbons right here, here, and here. So that's why it's called tertiary alcohol. But we cannot oxidate tertiary alcohol. So we need to change up the molecule somehow, and that's through isomerization, and we form isocitrate. So as we can see, this hydroxy group here is on the 3rd carbon. So first, second, the third right here, but on the isocitrate it's on the 2nd carbon, first, the 2nd, so we have it right here. So basically what happens is that we switch the places of the hydroxy group and we move it to the 2nd carbon and we get isocitrate. Now that we can oxidate isocitrate, we will in the process of oxidative decarboxylation. The word decarboxylation tells us that we are getting rid of the carboxy group, or rather CO2 or carbon dioxide, and oxidative that we are oxidating the isocitrate. Here comes NAD plus and it forms NADH. It tears away the hydrogen right from here, and we should remember this molecule because it will be very important. And the final product is called 2-oxoglutarate. Alright, so now we have the 2nd oxidative decarboxylation, and it's partly enabled thanks to coenzyme A right here. And again, here comes the NAD plus and forms NADH, taking this hydrogen from the coenzyme A, and the coenzyme A gets attached right here, forming succinyl coenzyme A. And we are also getting rid of another CO2 group right here. So CO2 goes away. Now we are left with the molecule of succinyl coenzyme A, which has 4 carbons in its molecule. So quick recap. First of all, we've added 2 molecules of carbon, one here, and we get the citrate, which had 6 carbons. Then in the process of first oxidative decarboxylation, we got rid of 1 carbon, and here we got rid of the 2nd carbon, forming the succinyl coenzyme A. Right, the next process is called phosphorylation. And basically what happens, as we can see, I haven't drawn the bond here normal way. It isn't straight, it's like a bit wavy, and that's symbolizing macroergic bond, the bond which has basically more energy than the other bonds, and this energy is used to form ATP from ADP. So first we have ADP, which is adenosine diphosphate, and thanks to this energy from the macroergic bond, it attaches 1 phosphate to the structure, and we are left with adenosine triphosphate, so ATP. And the product of it is succinate. So maybe one more thing to know is that this reaction right here doesn't form ATP straight away. It goes through GTP, which is guanosine triphosphate. It's a very similar molecule, but instead of adenine we have guanine in the structure. So first we got GDP, it's guanosine diphosphate. Through this macroergic bond, which energy is used to attach one phosphate to this molecule, we gain GTP. And from this GTP, the phosphate is again torn apart. So we get again GDP, and the energy that we gain by subtracting the phosphate is used to form ATP from ADP, which we got right here. Now comes another dehydrogenation. So basically, as we have these hydrogens attached to these carbons, what happens is that FAD comes. FAD stands for flavin adenine dinucleotide, and it forms FADH2. So we're stealing one hydrogen from here and one hydrogen from here, forming this double bond in fumarate, which is the product. We also should be aware that fumarate, because it has a double bond, we have to consider its conformation, which is trans, because it has these two carboxy groups on the opposite side of the double bond. Next process is hydrotation, and basically what happens is that we are getting rid of the double bond in fumarate by adding H2O, so that's why it's called hydrotation. So here we got rid of the double bond, and here we've attached the hydroxy group here. And the last reaction is again dehydrogenation. So we have to get rid of this hydrogen right here through NAD plus, forming NADH. And we are finally back to oxyl acetate, which was the first substance that acetyl coenzyme A reacted with. And it's ready to get back into the cycle and again react with acetyl coenzyme A. And this cycle goes indefinitely in the matrix of mitochondria again and again and again. So now we should summarize the outcome of Krebs cycle. Basically by forming 6-carbon molecule out of 4-carbon molecule, and in the process getting rid of those carbons twice, we are able to get NADH 3 times. I mark it here in red, so we have 1, 2, 3 NADH. Then we are getting one molecule of ATP, which is basically like energy currency in the cell. So cell uses it for various purposes when the reaction needs energy. And also we are getting one molecule of FADH2 that we will also talk about later.